Sampling of some of the work we're doing in the cancer stem cell arena. As an antibody company, we're focused mostly on cell surface targets that might allow us to inhibit cancer stem cells. And I'll tell you about some of the models and some of the approaches that we're taking, as well as some of the arsenal at our disposal at Medimmune that allows us to use antibodies as therapeutics. I'll also tell you about the approaches that we're taking that involve not only trying to find new targets for cancer stem cells, but also test some of our current agents for cancer stem cell activity. There are still quite a few challenges in this field, as we all know, and so I'll close my talk by just raising some of the questions that have also been raised by some of the other speakers that represent some of the challenges that we're still facing in developing drugs against cancer stem cells. So previous speakers have already told you about the properties of cancer stem cells, so I will not belabor this slide. However, one thing I will point out, because I think it will be important in some of the subsequent slides, is that the drug resistance properties of cancer stem cells means that when you treat with some chemotherapeutic agents, you actually see a proportionate increase or enrichment in cancer stem cells in the tumor population. And again, this is representative of the chemotherapeutic resistance that we see in cancer stem cells, and of course is one of the reasons that we're so interested in targeting this population, since they appear to be responsible for relapse and recurrence. So Dr. Frank is also taking you through this slide, so I'll also not belabor this. This is the now very frequently used review paper from Zhao et al. Describing approaches to targeting cancer stem cells with therapeutic agents. And again, there are various approaches to doing this. All of these are amenable to biologics approaches, including antibody approaches. And it's likely that it will be the combined effect of different agents that target cancer stem cells in the bulk tumor population that will really give the best results. In some cases, those combination effects will reside in a single agent. We saw that with the Oncomed DLL4 antibody targeting angiogenesis as well as inhibiting cancer stem cells. But there are also other agents. For example, Jenny Wang has suggested that, Jen Chang has suggested that lapatinib might actually have cancer stem cell inhibitory effects also. So again, there are different routes to developing protein therapeutics, or specifically antibodies that target cancer stem cells. We can certainly look for new targets, and we're doing that at Medimmune. In addition, there are the candidate targets that we have found from years of molecular biology that serve as potential targets for cancer stem cells, and these include things like the Wnt, the Notch, the Hedgehog pathway. But in addition, we want to be looking at some of our current agents. Not all of the agents that are being developed are resisted by cancer stem cells. Some of these agents are, in fact, dually functional in inhibiting not only the bulk tumor population against which they were originally designed to work, but also may have some cancer stem cell activities. So, of course, we have multiple approaches to doing this, multiple model systems and assays. Of course, the gold standard tumor initiation assay in mice is what we all rely on. And here's just one example of our pancreatic cancer models, where one model where we have, by FACS analysis, isolated the CD44 high, CD424 high population, and demonstrated that these can form spheres as well as in limited dilution, in this case 50 cells, can grow tumors in an immunocompromised mouse. In contrast, the population that is depleted for the cancer stem cell population